Thank
So
you.
our second speaker is Marcella Birtele from Georgia Quadrato's lab on almost exactly the other side of the planet, to tell you the truth, right? The West Coast instead of Europe. Um, and so I'll let you take it away. Thanks so much.
Thank you, Carlo. Can you see my slides?
Yes, we can.
Yeah. So yeah, it's exactly true going from the cold Finland to to Los Angeles. So hi everyone, and thank you, Carlo, for inviting me for this presentation. So I'm a postdoc in the Quadrato Lab here at USC, Los Angeles. And the focus of my postdoc has been to really try to understand the role of a top ASD-associated gene, which is SYNGAP1, in the context of human cortical neurogenesis. And so what we know about SYNGAP1 is mostly from work that has been performed in rodent models, where SYNGAP1 has been described as a protein with a distinct role at the postsynaptic level of excitatory neurons. And this role has been only recently shown to be equivalent also for human excitatory neurons in an iPSC model system where they generated adult human excitatory neurons. So when I started this project, we really wanted to understand SYNGAP in a more physiological context and how SYNGAP might play a role in other cell types. So we at first leveraged an already published data set, a single cell sequencing analysis of different developmental stages of human telencephalic tissue. And we looked at SYNGAP expression. So what we could first see was that SYNGAP expression was overall present at the different developmental stages, with an increase in expression at later developmental stages, in line with the findings of being a protein in excitatory neurons. However, when we looked at the expression in the different cell types, we were really surprised about the enrichment of SYNGAP in radial glia cells. And this was really something that was not described before. So what we wanted to do was to validate this data at the protein level. So what we did was to leverage a 2-month-old cortical organoid model that we generated with the Velasco, previously published Velasco protocol from the Arlotta lab. So what you can see here is an organoid with SOX2 positive cells, which are the progenitor cells that form these ventricle-like structures. And when labeling for SYNGAP1, we saw the expression of SYNGAP1 in the outer layers of this ventricular zone, which is typically the area where neurons migrate after the progenitors are proliferating and giving rise to neurons. But to our surprise was the expression of SYNGAP1 in the ventricle, specifically in the apical side of the ventricle, here highlighted in red in this high magnification. And it was actually co-labeling with TJP1, which is a tight junction protein known to have an essential role in maintaining the integrity of the ventricular wall. So we wanted to even confirm further the presence of SYNGAP in progenitors and human radial glia cells. So we looked at day 7 organoids where we have a homogeneous population of progenitor cells, and we performed the mass spectrometry in collaboration with the COBA lab here at USC. And what we could find was the expression and presence of different peptides from SYNGAP protein, and particularly the expression of the SYNGAP alpha-1 isoform, which is the isoform that was previously described to be essential and important for the excitatory neurons in rodent models. And by immunoprecipitating SYNGAP, we also confirmed the interaction with TJP1, as we have seen in the organoids. So now that we confirmed the presence of SYNGAP in radial glia cells, we really wanted to start to understand the function in this cell type. So at first, we performed the bulk RNA sequencing of organoids that were derived from a patient carrying a mutation in the SYNGAP gene. So this mutation is a point mutation in a specific in the RAS-GAP domain, which give rise to haploinsufficiency for the protein. And these patients have developmental delays, intellectual disability, epilepsy, and also autistic features. And what we did was to compare this patient-derived organoid with a control line, where we specifically CRISPR corrected the mutation to maintain the genetic background of the patient. And from a first look at the differentially expressed genes in the biological processes, we could find an enrichment in genes and terms for cytoskeletal remodeling, specifically cell motility and locomotion. And when looking at the interactors of SYNGAP after immunoprecipitation of the protein, we could also find an enrichment in proteins related to cytoskeleton, really hinting for a role of SYNGAP in the cytoskeletal remodeling in radial glia cells. And we know that cytoskeletal remodeling is really important in the context of the developing cortex. So here you can see a schematic where we have radial glia cells, which lays on the apical side of the ventricle, and they extend two cytoskeletal processes. One apical process where tight junction proteins are known to play an essential role in maintaining the integrity of the ventricle wall. And on the other side, the radial glia cells also extend a basal process here that is essential for the proper migration of neurons and their proper localization in the different layers. So to model this very early stage of the ventricle formation, we collaborated with Randy Ashton, who developed a 2.5D model system where we can culture cells in a confined area and differentiate them into neural progenitors. And so upon differentiation, these cells will spontaneously start to organize and to form a rosette-like structure, which resemble the ventricle formation of the brain. So here you see bright field images of this process where we can really see the formation of the ventricle and the rosette-like structure. So we use this model to assess first differences in the cytoskeletal remodeling between the patient line and the corrected one. So what we did was to look and quantify the percentage of single rosettes that were formed from these cells. And what we could find was that the patient line was really having an increased percentage of cells that were not able to make the rosette. And whenever for the small percentage where rosettes with the single ventricle were formed, the actual structure of the rosette was quite different, as you can appreciate from this representative image. So the perimeter was much larger, and also the organization was less structured. And so to even look more into the role of this of the mutation in the cytoskeletal remodeling of human radial glia, we used another control line called O323-1, and we inserted the mutation in this control background. So what we saw here was, similarly to the patient line, was also here in this case an increase in failed rosette formation. And whenever formed, they were also presenting a larger lumen and a more irregular, with a more irregular shape. So really highlighting the role of SYNGAP in the cytoskeletal remodeling, but also indicating that in this case, the genetic background of the patient did not play a role in this phenotype. And as I mentioned before, the mutation that is present for this patient is within the RASGAP domain of the protein. And so to really understand if this domain plays the important role in the cytoskeletal remodeling, we generated another line within this control background where we inserted mutation that resulted in homozygous RASGAP dead function, meaning that the RASGAP domain was not functional. And in this case, we actually could see that most of our cells were not able to generate single rosette, indicating that RASGAP might be the domain that is essential for this new function of SYNGAP in the radial glia cells. But of course, we're an organoid lab, so we wanted to validate these findings in our organoid system. So here in the upper panel, you see a 2-month-old organoid where we labeled neurons, deep layer and upper layer neurons together with intermediate progenitors. And what we can see normally in organoids is outside of this ventricle zone is a pseudo-layering of these neurons. However, when we look at the SYNGAP patient-derived organoids, you can really appreciate that this organization was completely lost with the presence of neurons already within the progenitor zone, so within the ventricle. So we continued our analysis in the ventricular zone by looking at how these cells divide, so how radial glia divide. And when comparing corrected with patient line, we could see that the corrected line would maintain a homogeneous ratio of self-renewing and differentiative divisions. However, when analyzing radial glia cells from the patient line laying around the ventricle, we could see that these radial glia cells were dividing more towards a differentiative divisions, suggesting that this impairment in radial glia scaffolding cytoskeletal remodeling affect the trajectory of radial glia cells. And at this point, so at 2-month-old organoids, we performed single cell sequencing from patient and control corrected organoids to have a more in-depth profile of these radial glia cells. So at this point, we found most of the cell types present in 2-month-old cortical organoids from both the lines. However, when we zoomed in the apical radial glia here in yellow, for the SYNGAP patient line, we could see a downregulation in the mitotic terms, and instead a really striking upregulation of maturation and synaptic markers for already within the apical radial glia, confirming our findings from the division mode and really highlighting the effect of SYNGAP haploinsufficiency in the division mode of radial glia cells. So next, we wanted to look at the overall composition of the organoids. And what we could see was that in control organoids, we had an increased level of progenitors. And at the same time, in patient organoids, we could see an increased level in overall neurons. Suggesting that this radial glia cytoskeletal remodeling is affected in a SYNGAP patient and giving rise to an asynchronous neurogenesis. And further, we collaborated with Gavin Romba, who shared with us a heterozygous and also a knockout model for SYNGAP, where we wanted to look at the phenotype that we observed in organoids. And interestingly, we found that both SOX2 thickness and new end thickness showed a similar trend compared to the one we saw in the organoids. So for SOX2, we saw a decrease in the head and even more striking decrease in the knockout. And similarly for the neurons, we could see an increased number of or increased neuronal thickness in the head and even more increase in the knockout model. And this is also in line with other findings in other models, such as the Xenopus, where they also found that SYNGAP haploinsufficiency alters the ratio between progenitors and neurons, indicating that this phenomena and this function for SYNGAP is shared and maintained across different species. And lastly, we wanted to look also how this early phenotype would affect later stages of the development and maturation. So we performed single-cell RNA sequencing in 4-month-old organoids. And here you can see highlighted terms for corticofugal and cortical projection neurons, where we could see that in patient we had an upregulation of different maturation markers, suggesting that, again, SYNGAP accelerate neuronal maturation. And we also validated these single-cell results by doing and performing a structural analysis that I'm not showing here, but where we saw an increase in the dendritic arborization of patient-derived cells. And also when looking at calcium imaging, we could see that both the patient line showed an increase here in gray in activations, in calcium activation over time, but also when looking at the mutation inserted in the corrected or the wild type line, we could see a similar phenomena of accelerated maturation. So to conclude this work, we really found a new function for SYNGAP in regulating human cortical neurogenesis. And this work reframes our understanding of well-known synaptic proteins and how these proteins might play a role at different stages, so specifically in early development. And this calls for investigating not only SYNGAP but also other top ASD-associated risk genes in the category of synaptic protein, and to understand and look at their role in different cell types and at different developmental stages. So what we're currently doing now in the lab after all this work on that patient line, we want to increase our number and observation for SYNGAP variants. So we have different lines now in culture from patients, and we are performing a first screening with the Rosetta assays to really establish how different variants might affect in a different way the cytoskeletal remodeling properties of radial glia cells. And with this, I want to thank everyone, especially Giorgia Quadrato for her mentorship, Ashley Del Dosso, who's a co-author with me in this study, and all the people and funding involved for this study. And thank you for listening. I'm happy to take any question.
Thank you. Please do jump in with questions. So I think it's very cool. Oh, Helena, jump in, please.
Hey, can you hear me okay?
Yes, we can.
Just that, thank you. That was fascinating. I think this this kind of accelerated maturation phenotype is now kind of popping up a little bit here and there, and of course your focus here was a synaptic gene. Have you looked at any other type of autism associated genes in the group perhaps yet? Have you seen something similar with genes in certain other categories than sort of synaptic genes?
Yeah, no, so this work essentially in the lab, we have been focusing first with the SYNGAP. However, now, with the interactor analysis, so whenever we pull down SYNGAP, we saw different interactors that are in the cytoskeletal, let's say, category, but they are also top ASD-associated gene. So one new project in the lab is also to investigate how we could have a hub of different top ASD-associated gene with a similar function in cytoskeletal remodeling at early stages. But it's difficult to gather lines from patients. So we're now thinking more of an approach with CRISPR to look at different genes.
Okay.
Thank you.
Cool. Thank you.
I think it's very important that work like this exists that is not predicated on a preconception of where and when this gene should be working. I think all of us are pretty aware that, once a protein product is seen in a synapse, a lot of the time and dollars that go into studying that gene end up way down the road from where you're looking, right? So none of this would be viewable if you had just thought about that. Silky, jump in.
Hi, that was great, very interesting result. I have two questions. One is first, have you looked at earlier phenotypes since like SYNGAP? This gene expression, it's kind of expressed even from the pluripotent cell, and gene expression is kind of steady, right? So I'm kind of wondering before you actually see the neural rosette formation, rosette-like structure exists in the earlier stage too, right? So whether you are aware of this one, have you explored those one for maintaining of pluripotency is different or something like that? And second question is also with the patient line, knockout line, besides the neuronal population, accelerated neuron, have you looked at other glial cell population or outer radial glial cell proportional changes in this cell line? Or yes.
Yeah, thank you. Yeah, so for the first question, at earlier stages, so we want to look for example, in neuroepithelial cells, if we already see an effect, of this haploinsufficiency. We don't have results yet, but yeah, that's something we would like to do. And I didn't quite understand what you meant at the iPSC's state. But yeah, so the only thing we have in plan is to look at the neuroepithelial cells. And then, yeah, for the second question, could you just repeat it?
Yes, for the outer radial glial cell or glial population, right?
Yeah. So we actually looked at intermediate progenitors. We didn't see any difference in the, for example, TBR2 positive cells. And also we haven't focused on outer radial glia cells yet, but in the lab we have a project where we're doing lineage tracing to really dissect, the possible, let's say, differentiation or pathways that are affected. Because what we felt was we tried to do stainings and look at outer radial glia. However, it's really difficult, in the cortical organoid model to really dissect how different proportion of apical or outer radial glia cells behave over time. So that's why we're going into the route of lineage tracing to have a better and clearer view over time of this phenomenon.
Can I ask a question, Carlo? So I find myself really interested in this story. Thanks for presenting it so easily to understand. And the part where I may have blanked out on this, but didn't you show us an activity measurement at the end?
Yeah.
Like some kind of calcium imaging, maybe.
Yes.
So then I was wondering about this relationship between these different functions of SYNGAP1. So the way I understand it is that SYNGAP1, along with TAM kinase 2 alpha and PSD95, are amongst the most abundant proteins in the brain. And that's how these genes were first isolated, because the proteins you just cut them out of a Kumasi gel and sequenced them. And so the point, though, is that that's because they're postsynaptic. So these are polarized machines, and you made that point that it's apical right at the beginning.
Yeah,
that you see this rather interesting sort of apical localization. At the time, I think, when it's not entirely clear to me whether those cells that are expressing it at that point are actually radial. They're radial glia, right? And they're stretching. They have a process stretching right across the epithelial sheet. So that's really a representation of the polarized distribution of SYNGAP1 within that epithelial cell. Sorry to make it a bit crude, a bit complicated. But anyway, the issue sort of is that Willesee et al. In one of these first autism papers did a whole study in Xenopus, which actually you reference.
Yeah, yeah.
And they showed the mutations in SYNGAP1 reduced telencephalon size. And I remember when that paper came out, there's a couple of interesting things about it, but one was that neurexin-1 mutations had the opposite effect. So a presynaptic mutation kind of had the opposite effect. And so then I was thinking about your imaging and of activity, and thinking that it'd be very interesting to ask at a high resolution level what's happening with synapses at that very first point when I assume circuits are forming. I don't know if that's a question or a request or whatever it is, but it seems like an interesting place to look very hard.
Yeah, it's absolutely interesting. So the thing is, it's very difficult to decouple, different stages and different roles that SYNGAP or other proteins might have, during the different stages. So that's why we wanted at first to look at the specific cytoskeletal remodeling function within 2.5D homogeneous, radial glia cells population. So with that assay, we kind of saw that already at that stage, there is an impairment in the cytoskeletal remodeling. So what we think is that, after with the problem in the remodeling, then there is kind of a cascade of, let's say, issues within the development of, and generation of neurons. Because as I mentioned, these cells really have the basal process that is important also for migration. So we see actually neurons in the organoids that are present within this progenitor zone. So we think that, these neurons are kind of stuck and they're not able to even migrate, to the proper layer. And that can cause miswiring.
Can I ask you a question? Do you think there's a neurotransmitter involved here? I mean, how far can we push the idea that it's a different function?
Yeah,
seems to me to almost like pose the question, well, maybe it's very, very similar, and that it's not a sort of highly polarized synapse, but it's all cells are all epithelial cells are polarized.
Yeah,
and you're making me think very hard about your response to me. Look, we went early because we wanted to keep it relatively simple and understand the cell biology. Yeah, I agree. Yeah. But then the cell biology seems to suggest that apical surface is a postsynaptic density forming. And so then you ask, well, is that transducing information from the ventricle? And the answer to that is absolutely it is. That's Chris Walsh, amongst others, but beautiful work, showing, for example, that there are growth factors that are acting on that surface to stimulate the growth of the epithelial cells there, the neuroepithelial cells. It's very interesting.
Yeah, that.
What's surprising is that to relate to what Silke was asking is it is there earlier. All these things are there earlier, but for the most part, it looks like excitatory neurons in the adult brain, at least, they're highly enriched in excitatory neurons. And why that is, I suppose, is because the excitatory neuron is sort of designed to be a highly polarized information transmission system. I don't know, that might be a bit of a platitude, but it's very provoking. Thanks so much.
Thank you.